All right, so we've talked about a lot of stuff, and I want to talk about one more thing, which is reproductive cloning, which is making a clone, essentially forcing parthenogenesis or asexual reproduction in vertebrates or animals. And it has been done, it has not been done in humans, but it has been done in mammals. So how we do that? They take a cell, an egg cell. So this is the secondary oocytes gone through meiosis. It has not been fertilized, but the nucleus is haploid, okay? And it has all that extra cytoplasm that that cell needs. Then you take a nucleus out of another cell, a somatic cell, and insert that nucleus into the egg cell. Now, why is it important that it's a somatic cell? Because the somatic cell has a full set of DNA. Remember, this nucleus, the egg nucleus, is haploid. We need a diploid nucleus. So you take the haploid nucleus out, put the diploid nucleus in, and then you grow that in culture, just like when doing in vitro fertilization, which is where you mix egg and sperm in a petri dish and hope that some of them grow into a little blastocyst of cells. So you wait to see if they grow, and then you implant that into the uterus of a female. And so the first time this was done with mammals, they took a somatic cell from the same individual that created the egg and that they implanted it into. And so the first clone, Dolly the sheep, gave birth to a daughter who was her genetic identical twin, not twin, but who was genetically identical. So this, like I said, this has not been done with humans, but having done it with mammals shows that it could theoretically be done with humans. There are a lot of ethical considerations there. And when you create a clone, you create a new infant. You don't create an adult. And that clone is going to be, while it is genetically identical, it's going to have a whole different life experience starting in utero, in the womb. And so it is not going to end up being exactly the same individual as the parent. And it turned out that Dolly, the sheep, the first cloned sheep, she actually died at a very young age of diseases associated with old age, which probably has to do with the telomeres on her chromosomes. So there's a lot that we don't understand yet. And there's a lot of ethical considerations to think about before we do too much of this. Now, one thing that we can do is what we call therapeutic cloning, where we take embryonic stem cells from an embryo, and these are normally from embryos as a result of an abortion with the mother's permission. Okay, we don't take these without the permission of the mother. Because those embryonic stem cells haven't had most of the genes turned on and off, you can then manipulate which genes get turned on and off, and you can create specific cells. And so we've been able to make brain cells and heart cells in petri dishes. Now that's, again, a long way from making a whole individual, but we can grow certain types of tissues in a petri dish as a result of therapeutic cloning. One thing we can't grow, and we really can't figure out how to do it, is blood cells, red blood cells, because they don't have a nucleus. They go through this special extra process to develop into a cell without a nucleus. We have not figured out how to make them synthetically, and which is why it's really important, if you can give blood, to give blood, because there's no synthetic replacement for it. All right, so we had Dolly, the first clone. We have also cloned pigs for medical use, for things like organ donation. We've cloned, some people have tried to clone some endangered animals that are very closely genetically related to domestic animals like cows. These are all endangered kinds of cattle. And again, there is a lot of realistic, logistical, and ethical considerations to think about with those kinds of things. Now, we can't clone humans, but in 2016, there was one very interesting thing done where a child was born to 3 genetic parents. The biological mother carried an allele for something called Lee syndrome, which is a mitochondrial disease. You remember, mitochondria have their own chromosome. It affects the nervous system. It's a devastating disease, and the mother didn't want to pass it on. The first 2 children that this couple had died from Lee syndrome within the first few years of life. And obviously they didn't want to do that, have that happen again. And, that's heartbreaking. And so they went to some doctors, and some doctors tried a new thing. And what they did was they took an egg from the mother and an egg from another woman, a donor female, unrelated. They fertilized the donor female egg with the father's sperm. They fertilized both eggs with the father's sperm. And then they took the nucleus of both eggs out, and they put the mother's nucleus into the donor female's cell, so that the new cell had nuclear DNA from the mother and father, the couple who had tried previously to have children. But the mitochondria in the cell were from the donor female. And so genetically, that child has 3 biological parents. So that is an interesting, thing. There are a lot of people trying to alleviate certain kinds of human suffering with these kinds of technologies. Some of them work out, some of them don't. So that's all for that, and on to other topics.